Good morning, Prep 3. In this video, we are going to talk about the second type of cell division, which is meiosis division or meiotic division. Meiosis division or meiotic division. So please pay attention. Meiosis division or meiotic division, I can call that reduction division. And we will know later why we call this division by reduction division. Firstly, can you remember what's meant by mitosis division? Mitosis division takes place in somatic cell. And producing two new cells. Each one contains diploid number of chromosome, and we said that diploid number of chromosome means complete number of chromosome. In meiosis division, the division takes place by different way. And this type of division, meiosis division, takes place in reproductive cell. And producing four new cells. Each one contains haploid number of chromosomes, not diploid. Each one contains haploid number of chromosomes. So let's go to know what's meant by meiosis division. What is the definition of meiosis division? It is a type of division that occurs in reproductive cell, by which each cell divided into four new cells. Each one contains haploid number of chromosomes. Pay attention. When the cell contains haploid number of chromosome, the cell will called gamete. Such as sperm in male, ova in female. Okay, let's go to what happens in meiosis division. Phases of meiosis division. We have two stages here. Number one, first stage of meiosis division, and number two, second stage of meiosis division. First stage of meiosis division and second stage of meiosis division. Let's go to know what happens in the phases of meiosis division. These are the phases of meiosis division. We have here in the first stage of meiosis division, we have four phases. Number one, prophase, and I called it prophase 1 to can differentiate between mitotic and meiosis. In mitotic, we called prophase only. In meiosis, we called it prophase 1, as meiosis division has two stages. The first stage, we have four phases, which are prophase 1, metaphase 1, anaphase 1, and telophase 1. Let's start to the first phase, which is prophase 1. Can you see it in front of you? Prophase 1. Pay attention. In prophase 1, we have two steps, at the beginning of prophase and at the end of prophase. What happens in each case? At the beginning of prophase, the chromatin reticulum condenses and forming long, thin, double-strength chromosome, and this is similar to mitotic division. Pay attention again. At the beginning of prophase 1, At the beginning of prophase 1, chromatin reticulum condenses, forming long, thin, double strings chromosome. And this is like prophase in mitotic division. The difference here in the beginning of prophase 1 in meiosis division, the chromosomes are arranged in pairs called homologous pairs. Can you see the pairs? Yeah, the chromosomes arranged in pairs. This pair called homologous pair. This pair called homologous pair. Pay attention. Look at the picture of a homologous pair. Yeah, that is colored with red and blue. This is homologous pair of chromosome. This is homologous pair of chromosome. We knew that each chromosome consists of 2 chromatids. So the homologous pair consists of how many chromatids? Yeah, it consists of 4 chromatids. Here in homologous pair, we have 4 chromatids. And these 4 chromatids, or this homologous pair called tetrad. This homologous pair called tetrad. So when I ask you about the definition of tetrad, what's meant by tetrad? You said that it is a homologous pair of chromosome by which each pair consists of 4 chromatids. Each pair consists of 4 chromatids. This is called tetrad. I can say tetrad, and I can say homologous pair, and I can say homologous pair. Okay, so in prophase 1, at the beginning, the chromatin reticulum condense and forming long, thin, double strings chromosome, and the chromosomes are arranged in homologous pairs, which is called tetrad. This is at the beginning. What happens at the end? At the end of prophase 1, crossing over phenomena takes place, and we will know what's meant by crossing over phenomena later. Number 1, crossing over phenomena takes place at the end of prophase 1. And what happens else at the end of prophase 1? Disappearing of nucleolus and nuclear membrane, and this is like mitotic division. Disappearing of nucleolus and nuclear membrane. Formation of spindle fiber. Formation of spindle fiber. Connecting the homologous pair of chromosome or tetrad of chromosome with spindle fiber from centromere. Okay, again, what happens at the end of prophase 1? Number 1, crossing over phenomena takes place. Number 2, formation of spindle fiber. Number 3, connecting the homologous pair of chromosome with the spindle fiber from centromere. Number 4, disappearing of nucleolus and nuclear membrane. Okay, so we know what happens at the beginning of prophase 1 and what happens at the end of prophase 1. Now we will know what's meant by crossing over phenomena. We said that crossing over phenomena takes place at the end of prophase 1. So please pay attention to know what's meant by crossing over phenomena. Now pay attention. This is a crossing over phenomena. Crossing over phenomena takes place between the homologous pair, such as in figure 1. The crossing over phenomena takes place in homologous pair. In figure 2, in number 2, what happens? What can you see in the figure? Yeah, the inner chromatid start to exchange. The inner chromatid start to exchange a part of it. Again, what happens in number 2? As you can see, exchanging a part of inner chromatids. Exchanging a part of inner chromatids. Okay. Number 3, what happens? Yeah, we can see that the inner chromatid exchange a part together. Exchange a part together. Why? Why we need to exchange a part from during meiosis division in homologous pair of chromosome? Why we need to exchange a part from inner chromatid? Okay. Here we need to form a new arrangement of genetic traits, which is DNA. Here we need to form a new arrangement of genetic traits, which is DNA. Why we need to make a new arrangement of genetic traits? Yeah, to make variation of genetic traits among the members of the same species. Pay attention, if you don't have crossing over phenomena in meiosis division, all the humans have the same shape. All the humans have the same shape, have the same eye color, have the same hair color, have the same length, have the same anything, have the same shape. If we don't make crossing over phenomena, all the human will have the same shape, So we need crossing over phenomena to make variation of genetic traits among the members of the same species to be everyone have its own shape, like its parent. Okay. So, what is the definition of crossing over phenomena here? How can I define the crossing over phenomena? This is a process, or this is a phenomenon that takes place at the end of prophase 1, by which the inner chromatid exchange a part of it. By which exchanging a part of inner chromatid in homologous pair of chromosome. What's meant by crossing over phenomena? Yes, it is a phenomenon that takes place at the end of prophase 1, by which exchanging a part of inner chromatid to form a new arrangement in genetic traits. Okay, so who can tell me what is the importance of crossing over phenomena now? Yeah, it is very important. Yes, it works on the variation, the differences, the diversity. Okay, it works on the variation of genetic traits among the members of the same species. So what is the importance of crossing over phenomena? It works on the variation of genetic traits among the members of the same species. Okay? And it takes place at the end of prophase 1. And this is what happens in prophase 1 at the beginning and at the end. Now, what is the phase after prophase 1? Yes, number 2, metaphase 1. Metaphase 1, as we know, in metaphase, in mitotic division, the chromosomes are arranged at the cell equator. And this is the same like in mitosis, in metaphase 1, in meiosis division. What happens in metaphase 1 in meiosis division? The homologous pairs. Yeah, pay attention. Here we can't say the chromosomes only. Here we must say the homologous pair or the tetrad of chromosomes are arranged at the cell equator. Pay attention. Can you see the homologous pairs in metaphase 1 are arranged at the cell equator after making crossing over phenomena, after exchanging the inner part of chromatids. Okay, here we have the crossing over phenomena at many phases, which is metaphase 1, anaphase 1, telophase 1. Okay, so what happens in metaphase 1? Yes, the tetrad or the homologous pair of chromosomes are arranged at the cell equator. Okay. The third phase is called anaphase. What's meant by anaphase, or what happens in anaphase? The spindle fiber in anaphase, as you see, the spindle fiber starts to shrink. So every group of homologous pair move away from each other, and each group migrate toward the pole of the cell. Forming two groups of homologous pair. Or homologous chromosomes or tetrad. Each one migrate toward the pole of the cell. Okay, this is what happens in anaphase 1. At the last phase in telophase 1, and this is called the adverse phase for the prophase. Can you remember? Yes. In telophase 1, what happens in telophase 1? The spindle fiber disappear, as you see. Formation of two groups or two nuclei. Here. Yeah, appearing of nucleolus and nuclear membrane again. So it is called the adverse phase for prophase. This is the opposite phase of prophase. Why? Because in telophase 1, the nucleolus and nuclear membrane starts to appear again. Okay? So what happens in telophase 1 again? Formation of two nuclei, and formation of nucleolus again, appearing of nucleolus and nuclear membrane. Pay attention here, formation of two nuclei, two new cells. Each cell contains haploid number of chromosomes. As you see, each one contains haploid number of chromosomes, not diploid number of chromosomes. So this cell will called gamete. Okay, but we said in meiosis division we produce 4 new cells. Each one contains haploid number of chromosomes, and these are 2 new cells only. Why? Yes, this is the first stage of meiosis division. In the second stage of meiosis division, each cell from these new cells forming 2 new cells. So finally we can obtain, or we can get 4 new cells of gametes. Yes. Now we can know what is the importance of meiosis division. if I ask you about meiosis, the importance of meiosis division, you can say, yes, exactly, you can say formation of gamete. Formation of gamete, and we know that what's meant by gamete? Yes, gamete is a cell, contains haploid number of chromosome. Can you give me example about gamete? Yes, like a sperm in male and ova in female. Okay, so the importance of meiosis division is formation of gamete. Formation of gamete. Thank you. This is only the first stage in meiosis division. In the next video, we will talk about the second stage of meiosis division. The second stage of meiosis division takes place by mitotic way. Okay, and we will know in the next video, inshallah. Thanks and goodbye.